Okay. Anybody trying to get in? Hopefully, Ming will advise me if there's anyone else trying to get in. Hello, everyone. We're really happy to welcome and introduce Professor Boyden from the Department of Brain and Cognitive Science, Media Arts and Sciences, Biological Engineering, who is also a Howard Hughes Medical Institute investigator. He's here to give a talk titled Tools for Analyzing and Controlling Complex Biological Systems, and we're all very excited to hear more about the areas he introduced in his abstract, such as expansion microscopy, optogenetic tools, and measurement of signals through signal reporting islands. Thanks again for joining us.
Great to be here. So if you're like me, you're probably all zoomed out. So I deliberately prepared topics that I would tee up and give highlights for, but I'd love to have more of a discussion. And so the formal slides I prepared are about half an hour, although I have about 50 backup slides. So if people want to learn more about a specific topic, we can go into more detail. But because the tools we build are so broad in application, they're being used to study everything from Alzheimer's to climate change to COVID-19 to you name it. It'd be great to kind of maybe at the half-hour point to get some feedback from you all about what you're most interested in. And then we can talk more about those things. So my group at MIT works in ways to analyze and control complex biological systems. My day job is I'm interested in the brain. And the brain, of course, has nanoscale building blocks like proteins and other gene products, as well as nanoscale anatomy, axons and dendrites of neurons. But brains are also large-scale systems. A single brain cell in our head might be many centimeters in spatial extent, the largest cells in our body by far. So how do you understand something that crosses so many different spatial scales? And the time dynamics too. High-speed signals are inside cells. And yet, long-term things like aging or Alzheimer's, they can take years or decades. So I sometimes say the problem of modern biology is space and time. Spatial scales, whether you study the immune system or a cancer or aging or development or climate change or biomanufacturing, these all involve vastly differing spatial scales and vastly differing temporal scales. So today I want to tell you 3 short stories about tools we've been developing to see and control biological systems across different spatial and temporal scales. The first topic is expansion microscopy. It's a way of doing nanoimaging of large 3D objects. Now, many methods for nanoimaging have been developed: cryo-EM, X-ray crystallography, super-resolution light microscopy, but all of them struggle to image large 3D objects like brains. So starting with 2 then grad students, and now about half my group works on this, Fei Chen and Paul Tilburg were the first 2 to work on this in my group. We thought, what if we do the opposite of what people have done for 300 years? Rather than zoom in, let's blow the biological specimen up. And so the way we do that is we weave a dense spiderweb-like mesh of swallowable material inside and outside cells, in between and around biomolecules. Anchor key biomolecules to the swallowable polymer, soften it, add water, and as the swallowable polymer grows, it'll pull the biomolecules apart from each other. So we call this expansion microscopy. We reported the discovery that we could swell a biological system in 2015. In panel B is a piece of the mouse brain. In panel C is the same piece of mouse brain, about a day and a half later. And as you can see, we've blown it up by about 100-fold in volume, about 4.5-fold in each direction. The polymer starts out very dense, like in the upper left, and it ends up extended, like in the lower left. Here's a time lapse of a brain being expanded. It's sped up from an hour to about a minute. And we add water right there. I hope you can see this brain is growing before your very eyes. So there are all sorts of things you can see with expansion microscopy that are difficult, if not impossible, to see with earlier means. One common strategy is to use fluorescent antibodies to stain for biomolecules like proteins. Here, in work led by Ray Gao, Sho Asano, and Gokul Padiyula, working with my group and Eric Betzig's group, we labeled mitochondrial and lysosomal proteins, shown in the upper right, myelin, shown in the lower left, and we can see these nanoscale features with ordinary microscopes. In other words, we democratized nanoimaging. Almost all biologists can access an ordinary microscope, but they don't see very well. Now we've upgraded everyone to make them into a nanoimaging device. Brain mapping. Could we make a map of the brain so detailed that we could understand how information flows? Well, you could express fluorescent proteins in brain cells, like shown at the bottom. If you zoom in, as shown in the top middle, you quickly hit the resolution limit of the microscope. But as shown in the upper right, which is the same field of view as the top middle, if you expand first, now you can see the fine wiring of the brain. Everything I've shown you so far is being imaged with proteins being visualized, but you can visualize other kinds of biomolecule too. Here, for example, we are labeling in work co-led by Oz Wassi and Fei Chen. We're labeling RNAs with in situ hybridization probes. This lets you see individual RNAs and, as you can see here, proteins within dense, intact brain circuitry. How big can you expand? Well, when J.B. Chang was a postdoc in the group, we've tried to expand biological specimens twice. And now in work that Deblina Sarkar, Jinyan Kang, Oz Wassi, and Margaret Schroeder have been spearheading, we've been figuring out ways to do this with all off-the-shelf chemicals. So here's a piece of brain. We form a polymer and expand a piece of brain. Form a second polymer in the space opened up by the first expansion, and you can expand it again. So now you can get extremely fine resolution. And furthermore, you can decrowd biomolecules from each other. You can separate them from each other. That's what you see at the top in the cartoon. Now we can see very fine features in the brain, like synapses, shown at the bottom. And the decrowding lets us get more accurate images than previously possible. So far, everything I showed you was in the brain. But what about other tissue types? Well, when Yongxin Zhao and Octavia Bucura were in my group, and in Andy Beck's group, respectively, we showed that we could expand a wide variety of human tissues: prostate, lung, breast, pancreas, the list goes on and on. Left is normal, right is cancer-containing. The color code is shown in the upper right. So now we can try to confront deep problems in medicine, like can you detect a disease early? Most diseases are hard to detect early because the changes are so small. But by expansion, we can make those small changes big and obvious. This slide is kind of dense and not really meant to be read, but probably about 1,000 groups all over the world are doing some kind of expansion. Over 195 papers and preprints have come out doing expansion, applying it to everything from the kidney, to the fruit fly brain, to transgenic mice, to looking at fragmentation of the genome, and the list goes on and on and on. We did hands-on training of perhaps 400 groups, but post-COVID, we can't do that anymore. So we have step-by-step cookbook-style protocols on our website, expansionmicroscopy.org. On the website, you can see how to do expansion with all off-the-shelf chemicals. And we even include detailed step-by-step photography so that people know how to handle the expanded specimens. So to summarize the first short story, we've discovered that we can expand evenly biological systems, not living ones, but preserved ones. That lets you do nanoimaging on regular microscopes. And it's a very simple method using all off-the-shelf chemicals. So please give it a try. We're happy to help with anything. Biology and medicine are fundamentally nanoscale sciences, right? All the building blocks of life are nanoscale. But up till now, there wasn't really an easy way for anybody to do nanoimaging. But now anybody can do nanoimaging. That's short story number one. Now, as must be all too obvious, expansion does not work on living things. It only expands molecules away from each other in non-living things. So the next two short stories I want to tell you about are about living things. How do we control and observe high-speed physiology in living things? The first story I will tell you about is optogenetics, a way to control neural activity using light. This started when Carl Deisseroth and I were both students at Stanford, and we just started brainstorming through all the laws of physics. How can you deliver energy into the brain and control brain activity? Brain cells have electrical activities that cause them to be active or silent. So we thought light could be very valuable as a way of controlling them. Light, after all, could be brought into the brain quite easily. And so the next question was, how do you make brain cells sense light? Well, I got very fascinated by a class of molecules from the natural world, so-called opsins from microbes. These are 7 transmembrane proteins, such as shown in the lower left, that occur in microbes, such as that live in salty water, like in the upper left. In 1971, light-driven proton pumps were found. 10 years later, light-driven chloride pumps were found. And around the turn of the millennium, light-driven ion channels were found. These are all from single-celled microbes. Flash forward to the current day. We now know that members of all 3 of these classes can be found that are fast enough, safe enough, and effective enough to mediate delicate neural control. After all, neurons are complicated cells. When Brian Chow and Shua Han were working with me, they found that light-driven proton pumps could be identified, that when genetically expressed in brain cells, you could shine green or yellow light on them and turn them off. That way you can figure out what those brain cells are needed for. In the middle of the slide, Shua and then also Amy Truong found that we could do something similar with light-driven chloride pumps. And finally, on the right-hand side, originally Carl Deisseroth and I worked on this, and then later Nathan Klepecki, Youngho Cho, or Shumesh, Valerius Impini, Dimitri Temnese, and others worked to bring these to their physical limits. We found that light-driven ion channels could be found that could be genetically expressed in neurons and then used to activate the neurons. Originally, as you can see from the color codes of these figures, the visible light colors used were green, yellow, and blue, for example. But what we worked on was to try to make these molecules reach their physical limits of performance. So for example, Amy Truong, when she was a grad student in my group, worked on having red light turn neurons off. Red light, after all, goes deeper in the brain and body than other colors of visible light. And Nathan Klepecki worked on a light-driven ion channel that could be driven by red light. These molecules have become popular for mediating red and even infrared light control of neurons. We also wanted to make the molecules very, very focal. One of the things that we've been working on is to express these molecules at the cell body so that they can be used to activate individual neurons or silence individual neurons, even that when they're embedded in a dense neural circuit. These molecules, known as optogenetic molecules, opto for light and genetic because these are encoded by genes, are very popular. We've sent them to literally thousands of researchers at this point. And so I didn't really prepare a whole lot about optogenetics, but I wanted to mention that we were very lucky. Optogenetics worked out of the box, basically. The molecules were safe enough, fast enough, and effective enough that we could take them out of the microbes and put them into the neurons, and they would work. It was great serendipity. And so I want to spend the rest of the talk talking about some new areas that we've been working on, including some very recent work about the opposite problem. With optogenetics, we control neural signals. But what about reading out neural signals? And beyond, right? There are 30,000 genes in the genome. What if you could monitor all 30,000 and what they do in the living cell? Well, of course, we haven't been so lucky. Whereas optogenetics was basically solved by the natural world, to look at cellular activities, we must invent new indicators. When Erica Jung and Kirill Pyakovich were in my group as postdocs, they worked on a project to build a robot that could do evolution. So we could take a gene, make millions of mutants. Some of the mutants are better and some are worse for any given goal. Put those mutants into cultured cells, as shown in this cartoon, and use a robotic microscope to image and extract the cells that have the genes of interest, as shown at the bottom half of the cartoon. We decided to try to make a fluorescent voltage indicator, a molecule that would light up when a cell was active in the brain. We did 2 rounds of robotic directed evolution involving 10 million mutants. We used a robotic arm to pick out the cells that were better, that had gene products that were brighter and better localized and more stable, as shown on the right-hand side of the slide. Using this strategy, we found molecules that were indeed better localized, as shown in the upper left, better in their signal to noise, as shown in the upper right, fast, as shown in the lower left, and stable, as shown in the lower right. Collaborating with Shu Han's group at BU, Kirill Pyakovich, Seth Bensussen, and Juan Sang worked together to use this molecule, which we call Archon, in the living mouse brain. We use a simple microscope shown in the upper left. We genetically express this molecule, which we call Archon, in neurons in different brain regions. And as you can see, we can look at neural activity in multiple brain regions, the cortex, the striatum, and the hippocampus. Because it's imaging, we could see many cells at once. Here are 8 cells active on the right from the mouse brain in an awake behaving mouse. Now, lots and lots of fluorescent indicators have been made. Literally hundreds are now available. Now, there are 30,000-ish genes in the human genome. The gene products work together in very complex networks. How can we understand all of them? These are some screenshots from Biosensor DB, a website at UC San Diego. It lists some of the hundreds of fluorescent biosensors that are out there. There are molecules that will light up in a living cell for changes in pH, ammonium ions, ATP, cyclic AMP. The list goes on and on and on. Ideally, we could image many of these at the same time in a living cell. Then we could figure out how these signals talk to each other. But this is difficult to do. Traditionally, you would use indicators of different colors. Have a green sensor of ATP, for example, and a red sensor for, I don't know, zinc. Then you could figure out how these signals relate to each other. But what if you want to do more than 2 colors? What if there is no sensor with the right color for you? Well, Shannon Johnson and Chengyang Lingu in my group recently showed that we could do a very different way of imaging many signals in a living cell. Here's the idea, shown in the upper left-hand side of the slide. Suppose you have a bunch of different fluorescent reporters of different signals, some of the hundreds that I mentioned on the previous slide. What if you could put them at different points in the cell? For example, the points labeled 1 might report signal 1, and the points labeled 2 might report signal 2. While the cell is alive, they'll be blinking at you, reporting their signals. If they're at different points in space, then you can tell the signals apart, even if they're all the same color. But what if you need to figure out which signal is at which point? Well, once you're done, you can preserve the cell and then use staining to figure out what signals were at each point. For example, as I told you at the beginning of the talk, expansion microscopy is a very powerful technique for enabling staining of molecules, right? We could apply a stain, take a picture, wash it out, apply a second stain, take a picture, wash it out. You can do this as many times as you want once the cell is preserved. Here's how we made it work. We would take a fluorescent reporter and attach it to a set of self-assembling peptides. Then these molecules will self-assemble into discrete dots within a living cell. Different reporters get different self-assembling peptides. We also fuse to each reporter an epitope, a small peptide that later you can stain, for example, with an antibody, to figure out the identity of each spot. So if different reporters have different epitopes, even if they're all green, let's say, when the cell is alive, later you can come in with an antibody bearing a red fluorophore and stain against epitope 1, take a picture of all of the signal 1, wash it out, bring in an antibody against epitope 2, figure out where all the signal 2 dots are, wash it out, and so on and so forth. Amazingly, this works. Here in panel A, we take GCAMP, a popular calcium indicator, and we fuse it to a pair of self-assembling peptides. We think that helps make the clusters bigger. Panel B shows regular GCAMP beautifully expressed throughout the cell. Panel C shows GCAMP in these puncta after it's been fused to the self-assembling peptides. We did lots and lots and lots of control experiments to show that the kinetics, the signal to noise, the dynamic range, the physiology of the cell, and so on and so forth were not altered by the clustering. Amazingly, this design was modular. We could take different fluorescent indicators and fuse them to different peptides, and they would then self-assemble in clusters in different points throughout the cell. So panel A shows a cultured neuron. On the left is just the anatomy. On the right are a bunch of green dots. These green dots are from 3 different clustered indicators: GCAMP6, a calcium indicator; CAMP-R, a cyclic AMP indicator; and X-ray ACAR, a pKa indicator. Some of you might know that these are some of the most important signals in all of biology. They're involved with everything from learning and memory to cancer metastasis and everything in between. But how do they signal to each other? How do they relate to each other? Can we decipher the network within the cell that makes the cell work? Panel B and C show zoom-ins into the upper right. The green images show the live image, green dots that, if this were a movie, you could see blinking at you. On the right-hand side of panel B and C are the colored images showing the post-hoc staining. Some of the green dots turn purple, meaning that's calcium. Some of the green dots turn blue, cyclic AMP, and some of the green dots turn yellow. That's protein kinase A. So what this means is, for the first time, even using a single color microscope, so it's very cheap, you can image 3 different signals in a living cell. And you can figure out how they relate to each other. Now, as I mentioned, we picked these 3 signal indicators because they're very important for learning and memory. A big question in neuroscience is, how does a short-term change become permanent? We all have memories from childhood. How does a 1-minute experience turn into something we can remember 10 or 20 or 30 years later? Well, in panel H and panel I on the right, you can see we saw different kinds of response of the neuron. Some in panel H had delayed fluctuating calcium responses to a cyclic AMP challenge using the drug forskolin. Some in panel I had a fast calcium response. How does this relate to the pKa output? Amazingly, we could predict the pKa output from the speed of the calcium response. In panels J, K, and L, you can see how this looks. Neurons with a fast calcium response, shown in red, had a stronger pKa response, shown in panel L. Neurons with delayed and weaker calcium responses, shown in blue, had a weaker pKa response. We could use these tools in the living mouse brain as well. Using these, we could show that we could analyze these signaling networks in the ex vivo brain slices from the mouse brain. And we found similar responses. Neurons with faster calcium responses had stronger protein kinase A responses. Neurons with weaker and delayed calcium responses had weaker protein kinase A responses. How many can we look at? Well, for fun, we took 4 different sensors, fused them to 4 different self-assembling peptides and 4 different epitopes for post-hoc staining, and showed that we could get 4 different sensors to cluster at once. That enabled us to see 4 different signals at once in a living cell. And then by adding a 5th reporter in, we showed we could see 5 different sensors at once. So to summarize this last part of the talk, we've been working on new tools for seeing cellular physiology. We can evolve new molecules using robots, evolving them to have better performance. We also can think about the use of tools in new ways. For decades, people have recorded multiple signals primarily by having them be different colors. But now we know we can also observe different signals from different points in space. Oops, I'm not sure why I jumped backwards so many slides. So that was the half an hour that I prepared. I acknowledge all the people along the way who led the projects, but this slide is very helpful just because there are so many people, both in the group at the top and around the world in the middle, who have collaborated with us on these projects. And with that, I would like to take questions, although I'll note that our website, synthneuro.org, has access to all of these tools and protocols for how to use them. We have a big culture of sharing. Expansion microscopy and optogenetics are already in use by probably over 1,000 groups each. The spatial multiplexing and voltage indicators are newer, but are starting to find lots of uses throughout biology. And with that, I'd be happy to take questions.
Thank you very much. I'll allow people to mute themselves so people can write a quick question on the Zoom chat if they feel like it. Thank you very much for the talk. It was these 3 tools. I'm sure you could give 3 or 4 talks on a single tool. There's a lot of information there.
Yes, and as I mentioned, I prepared about several dozen more supplementary slides if there's a topic that somebody wants to go into in more detail. But given the breadth of synthetic biology and the theme of your organization, I thought it'd be more fun to have a conversation about it than for me to lecture about something if it's not in somebody's interests.
Let's see if there's any question ready. Otherwise, I'll start shooting off myself. Yes, so if I could quickly ask, so when it comes to expansion microscopy, from what I understand, you're using handles to spread the molecules of interest apart. But in the absence of these molecular handles for everything else, can these other components be also imaged, or are they dispersed and it creates more difficulty?
Well, the molecules with the handles are the ones being pulled apart. If we don't have a handle, those molecules get washed out during expansion. But we have many handles. We can bind to proteins, RNA, DNA, and many people are inventing new handles all the time.
So just to clarify, so what you're looking at, what you're searching is the compounds you've attached the handles to. So that's the focus of the microscopy.
Well, yeah, I mean, light microscopy, you don't see the actual thing, you see the label you attach to the thing. So if you can label it, you kind of need to label it anyway in order to see it, right? So that's why it's so popular. It's completely compatible with how microscopists think.
And I have a follow-up question again. This might seem a little banal, but essentially with expansion microscopy, do you lose the relative disposition of molecules?
No, that's why it's useful. Yeah, so the expansions are very isotropic. The distortions are very small, maybe a few percent over a typical microscope field of view.
Even at the molecular level?
That's right. That's the whole point. Yeah. Sorry if that wasn't clear, but
No.
yes, it preserves things down to at least 20 nanometers.
That's pretty amazing. Thanks.
Yeah.
Let's let some other people ask some questions. Otherwise, I still have some more prepared myself.
Sure. Ask away.
Hello. Hi. Thank you for your talk. That's amazing. So I just have 2 quick questions. So the first one is for genetically expressing pumps within those neurons. So I know, like, will that affect the pumps that has been naturally expressed in the neurons? Will they
Nope.
like interfere with each other?
Not at all. No, the pumps we express are from different species, and they have no interaction that we know of with anything in the neuron. That's why they're interesting. If they did have an interaction, then they wouldn't be interesting.
Okay, and my second question is about the Alzheimer's diseases. So you mentioned that you can use this technology to kind of treat for Alzheimer's diseases, or maybe find the biomarkers for future drug development. Can you elaborate more about this point?
Sure. My collaborator, Li Wei Sai, used optogenetics to discover that driving the brain 40 times a second cleans up Alzheimer's in mice. Using that discovery, the teams went on to figure out that you could induce these 40 hertz oscillations through having people watch a movie. So we're now running clinical trials of movies for Alzheimer's.
Okay.
So it's early days, but hopefully we can make progress rapidly.
Yeah, thank you.
Sorry, thank you for the talk. I missed what you said. What do you do for these patients? Could you describe how you aim to clear Alzheimer's?
Sure. My collaborator, Li Wei Sai, used optogenetics to show that if you drive the brain 40 times a second with our tool, the Alzheimer's gets cleaned up. We went on under Liwei's leadership to show that you could cause the same 40 hertz brainwave by having the mice watch a flickering light or hear a clicking sound. So now we're running human trials with 40 hertz flickering lights and clicking sounds to see if we can help stop Alzheimer's.
Right. Okay. And that's to stop it, like a pause of Alzheimer's development rather than like clearing it or regenerating it.
Well, it clears it. Yeah, stopping means clearing, but it doesn't regenerate it.
Yep.
But if you don't clear it, you don't stop it.
Sure. Thank you.
Sorry,
As far as I know, anyway. This is probably more a question for Liwei than me. I just make stuff.
Yeah. Thank you. I just have a follow-up question. So,
Ask away.
do you know why the like movie can stop the Alzheimer development?
Say that again, please.
Do you know what's the reason of like using the songs and the movie to stimulate the patient's brain to stop the Alzheimer diseases? What's the reason behind that?
We're still investigating the mechanism. Yeah, it's mysterious, isn't it? But it's
Yeah.
pretty, yeah.
Thank you.
Wonderful questions. Yeah. Anybody else have questions or things you want to talk about?
I have another question, which is a little unrelated. So I saw that you brought up a lot of different websites where you have protocols and you try to foster collaborations with other labs. When it comes to data sets, which are important in synthetic biology, or maybe some data sets that you find particularly interesting, accessible, interesting, or that you use during your research, what particular data sets would those be?
Sorry, you faded out there for a second. Data sets of what?
Sorry, so essentially in synthetic biology, for example, when it comes to neuroscience, I think there's the Allen dataset, which which seems to be quite well used in research. Are there similar data sets for synthetic biology that are well used in maybe in fields that related to what you've been working on?
Well, that's a good question. I mean, we are building these molecular tools and we give them to thousands of researchers all over the world. They then go and get these data sets and, but that's kind of their business, not ours. Are you asking like how they deal with the data sets? I mean, they do whatever they usually do, I guess.
Yeah, but in particular, your research is more fundamental for the tools. You don't really work with data sets produced by other people, from what I understand.
No, well, we collaborate lots of people on acquired data sets. I mean, all the methods I mentioned can be used to acquire data. Are you asking what software we use for the data? I'm a little bit confused about the nature of the question, I guess.
Sorry, it's probably not really related. I'll open I'll open up to other people, but essentially the question was whether you were using personally some data sets, similar to how the websites you use to share protocols. Are there similar resources to share the data from experiments?
Oh, I see. Oh, yeah, absolutely. I mean, every one of our papers has a data statement and a code statement, and many of these things are freely available and from repositories. But most of our papers are technical, so I don't know how interesting the data is. Most people just want to get their own data.
Thank you.
Great question.
Sure, I got like a few questions that link together, if that's okay.
Absolutely, go for it.
Sure. So I'm interested in trying to find consciousness, basically, if we can. I was wondering if we're able to measure what goes in and out from like a neuron or a brain cell, and if we could like track that, then we're more likely to see if there was like a discrepancy and say like, that might be consciousness happening. So first silly question is that have we found like all the molecules or the possible physical things that are happening inside the brain? Like, is there like a molecule or something that we're not aware of, or is there like a percentage you can give? Firstly,
I think we know almost nothing about the brain. I mean, there are thousands and thousands of genes. We don't understand what they do. We don't know how many kinds of cell there are in the brain. We don't know how they connect to each other. I would say that if here's the Earth and here's the moon, and we're trying to go from the Earth to the moon, and landing on the moon is the equivalent of understanding what the nature of consciousness. We're probably about 3 inches off the ground.
Oh, right. So that's quite exciting to hear. But with your technology, it's the first time I'm hearing about it. So thank you for sharing tonight. Well, tonight in London anyway. So would you see that your technique would be the right technique to use to basically see what's going more inside like the molecular brains, or like, you know, see like what's going in and out? So, sorry if that makes sense.
That's why we built it. We built these tools to want to solve the brain.
Sure. Okay. Right. Okay. I'll look in more into it. Thank you.
Great. Yeah, in the coming years, I want to start applying all these tools together. And by combining them, can we start to solve brain circuits?
Right. I'll look forward to it. Thank you.
Great. Other questions or comments? These are all great questions.
Yeah, sorry, if nobody is going to ask other questions, I have another one.
Go for it. That's what I'm here for.
So for your third technology, it's like a spatial thing. You can visualize different positions within the cells. But for the second technology, it's more about like temporal thing, because you can control the expression of some pumps like temporally. So my question is, can you like actually leverage them together so we can like control things like temporally and spatially?
Oh, yeah, yeah. I mean, that's kind of what we're trying to do in the last project, right? You know, when we have different temporal reporters at different points in space, we're trying to combine them. Yeah. Did that answer your question? I wasn't quite sure what you meant by it. So.
Yeah, could you like give me some more details?
Like what? What kind of details do you want? I mean, I could talk for 3 or 4 years about this, so maybe knowing a little bit more about what you're interested in would be helpful.
How do you add the temporal part into your third technology?
Well, I don't know if I was clear, but in the last part of the talk, those signals are temporal signals, right? Different points in space are broadcasting different movies, right? Was that clear during that part of the talk?
Yeah, makes sense.
That's how we combine the space and the time in the same experiment, right?
Okay, yeah, thank you.
Also, once you're done with that experiment, you could do expansion microscopy and analyze with even more spatial detail after doing the time analysis, right?
Okay, yeah. Thank you.
Great. Great. Other questions or thoughts?
Hi, sorry. So I have a question. I'm not sure if I missed this during the presentation, but could you either reiterate or explain how you designed and generated the signaling reporter islands? Because I'm not quite sure about what was the process of how did you discover it, how did you design it, and what are the principles of designing these signaling reporter islands? So
yeah. So we fused the fluorescent reporters to self-assembling peptides. The self-assembling
yes.
peptides can cause them to form clusters.
Yeah, so I think I'll be interested to know like what are these self-assembling peptides exactly? And
Say it again.
what are these self-assembling peptides exactly?
They're bioorthogonal human-designed peptides like coil domains and polyhedron forming domains.
Okay.
Yeah.
Did you design it yourself
You go first.
or?
Sorry, you go first.
Sorry. So did you design them in your lab or how did you come about?
Oh, no. There are hundreds and hundreds and hundreds of these designed by groups over many, many, many years. Amy Keating, David Baker. I mean, there's a whole protein engineering community where people are trying to design self-assembling peptides, and we just synthesize them and use them.
And how do they anchor themselves in the cell? Because
They're self-assembling.
Okay, so I assume that they don't move about in the cell.
Exactly. They self-assemble and become so big that they don't diffuse, right?
Okay.
Suppose you put a leaf on a lake, it'll diffuse around, right? But suppose you put a 500-ton boat in the lake, it'll stay there, right?
Okay. Yeah, I can see that.
Good. Great question. Yeah. Any other questions?
Sorry. So you mentioned that you put like a lot of the reporters and also I'm talking about the technology. So it's like you have lots of fluorophores floating around in a cell, how come like it can't affect the physiology of the cells? Because like it definitely can affect the communications within the cell, right?
Well, the reporters could affect the physiology of the cell. All I'm saying is that when we cluster them, we're not changing it any more than when they're freely floating around, right?
Yeah.
But famously, anything, if you express it at too high a level, could kill a cell, right?
Yeah.
Yeah.
Okay.
So we compared our clustered reporters to the regular reporters.
Okay.
But if you're using any technology, you must do control experiments and show that it's not corrupting the cell, right?
Yeah. Okay. Thank you. Yeah.
Good. Wonderful question. Other questions?
Sorry, Ming?
Yeah, I don't have a question yet.
Sorry. So these techniques are really interesting, and for example, application for Alzheimer's, like that's something that was probably very hard to predict. Are there any other such applications that you're aware of? Maybe not necessarily in your lab, but in the wider community.
Oh yeah. I mean, our initial paper on light-controlled neurons is cited 4,000 times. It's in use by thousands of people. People are finding patterns that might clean up Parkinson's, that might help with anxiety reduction, that might help fight cancer. The list goes on and on and on and on and on. Yeah.
Were there any in particular that you found very interesting that were really hard to predict, starting off from the technology?
Sure. Yeah. I mean, a group at Stanford found a better target for deep brain stimulation for Parkinson's by using the technology. There are groups trying to figure out how to shut down epileptic seizures. People are finding neural pathways that you could excite to turn off anxiety. People are finding neural pathways that you can use to turn off stress. Oh, the list. People are finding neurons that you can drive and they can cause you to recall a memory. These are all in mice, of course, but still, it's pretty cool. Yeah, the list goes on and on and on. I mean, literally thousands of people are doing this kind of work now.
Thank you. That's amazing. Are there any more questions? Seems we're winding down a little bit here, but in that case, I'll probably ask the final question, since unless another one appears before I finish. So essentially, these technologies are really incredible and groundbreaking. What do you see next for work in your lab? Are you going to be working on additional technologies? What's the groundbreaking aspect? What's the edge of the field as you see it?
Oh yeah, well, I want to see everything and control everything. So yes, we have a lot of things to do. My group has over 50 people in it, so we're working on approximately 50 different projects. And yeah, the next couple of years are going to see huge revolutions, I think, in how we see and control biological systems, because many groups now are starting to work in such things. So yeah, stay tuned.
Thank you very much. Do you have any more questions?
Well, check out our website if you want to use any of our tools. And if you need any help, don't hesitate to email me. My email is just edboyden@mit.edu. In addition to being the professor running the group, I'm also the chief technical support person. So if you need any help, just drop me a line.
Thank you very much. It's been amazing having you, and very eye-opening. I'm sure everybody really took home something now, and we're really excited to look into this a little bit more. And we'll definitely stay tuned in. Thank you.
Great. Stay in touch. Always happy to help.
All right. All the best.
Thank you.
Have a good rest of your day. Bye-bye.
You as well. Take care.